Lesson 3 on cell organelles. Cell organelles are considered tiny organs. They're the parts that basically do different functions for the cell. So they generally have outer membranes. That's how they're known because they separate themselves from the rest of the cell. But each of them performs a specific function. The cytoplasm is the portion that is inside the cell membrane. So here's our cell membrane. It's inside the cell membrane but outside the nucleus. Here's our nucleus. So everything in between would be considered the cytoplasm. Now when I think of the cytoplasm, I generally think of the cytosol because the cytosol does not include the organelles. The cytoplasm, if you read the actual definition, it says it does include the organelles except the nucleus. The cytoplasm, the purpose of that is for movement within the cell so that things can kind of move around within the space. Cell membrane is that outer surface, covers a cell's surface. It's also known as the plasma membrane. We'll probably refer more to cell membrane than anything. The function of it is to act as a barrier between the inside and outside of the cell. Anything that goes in or out of the cell has to go through the cell membrane. As we look up here, we know that we have the phospholipid bilayer. We talked about phospholipids back when we talked about lipids in the macromolecule unit. You can see all of the individual phospholipids that make up the layer. And if you remember from that unit, we talked about how the phosphate end, the circular end here, is hydrophilic. So it loves water. So water can exist over here. And water can also exist over here because this side's also hydrophilic. But in between we have a hydrophobic layer which hates water. So because there's that difference between water loving and water hating, things cannot easily go through the cell. They are either stuck on one side or stuck on the other side. And that is why we have things like protein channels, this here, that allows for a passageway to send things in and out of the cell that would not normally be able to pass through the cell membrane itself. We say that the cell membrane follows a fluid mosaic model, and what that means is that it's more liquid than solid. So I have a quick video here that I'll show you part of. So we see our phospholipids, and notice how they're kind of moving, flowing. They're not staying in one place. But this is a computer model of what we feel exists for the cell membrane. And you'll notice the protein channels are in there, and that allows things to go in and out. We'll talk about that process more in the next unit when we talk about cell transport. Okay. And before we move on, I do want you to circle a pair of the phospholipids that make up the inside and the outside of the cell. All right. The cell wall, that is plants only. The cell wall is a rigid layer that lies outside of the cell membrane. So plants have the cell membrane as well, but they also have the cell wall. And the purpose of the cell wall is to help provide support and function. It's made of cellulose. That is one of the carbohydrates that we talked about in the macromolecule unit as well. There are pores in the cell wall that allow things to flow in and out. So the cell wall it's not the barrier. The cell membrane is the barrier, and that's why we have the pores to allow things to enter and exit through the cell wall so that it's not an extra barrier, but it provides the support and protection so that those plants can grow tall and strong. So the other thing I want you to do is to recolor your image on your notes to identify between the plasma membrane and the cell wall, because it's not always obvious, especially if you're looking from a top down. We would say that the cell wall is this outermost layer, the green, and then this orange would be our cell membrane or plasma membrane. So I do want you to recolor the cell wall on your notes so that you know which item it is, just to differentiate between those two. The nucleus is what controls pretty much everything in eukaryotic cells. It is not in existence in prokaryotes. The prokaryotes don't have a nucleus, they have a nucleoid, which is the area where the genetic information exists. Now, in the nucleus, it stores the genetic information, the DNA, or deoxyribonucleic acid. Inside of the nucleus, so this whole thing is the nucleus. Notice there's nuclear pores that allow things to go in and out. Chromatin is a thread like DNA that is not condensed. So if we look down, whoops. So our chromatin is going to be more stretched out. The chromosome, we see a chromosome here, is condensed DNA that is organized around a protein. So when you take the individual strands, now this gets it down to the DNA and the gene, but when we get the strands of it, that would be the chromatin. When it's condensed, it becomes a chromosome, so it's all wound up. And the nucleolus, which is the center area, is a dense region of DNA where it makes the RNA. And we'll get into more of the DNA replication, which is a huge part of the nucleus and other parts that we'll be talking about that work together to get that duplication process going. I'm not going to go into a lot of the details there. The endoplasmic reticulum is a system of membranous tubes and sacs called cisternae, and those allow for transport. So think of these kind of like a highway. They transport molecules from one area of the cell to another. They do not go throughout the entire, like the endoplasmic reticulum itself is near the nucleus, and it just sends things to different parts of the cell. It doesn't go all the way to the cell membrane. There are two types. There's the smooth ER and the rough ER. You'll see the smooth over here. You'll see the rough over here. The difference is that the rough has ribosomes, the smooth does not. As far as the rough ER, I already mentioned there's ribosomes on them. You can see all the little polka dots. The function is responsible for producing phospholipids and proteins, and those phospholipids are going to help with your cell membrane. The proteins are going to help with multiple different functions, and they're exported from the cell or inserted into the cell membrane. So if you think about phospholipid membrane, it makes sense. Smooth ER does not have ribosomes. It looks smooth. It's responsible for building lipids like cholesterol, so not the ones that go into the cell membrane. Releases calcium in the heart to stimulate contraction, which is used in cardiac cells. Produces estrogen and testosterone, which get used by the reproductive organs. Detoxifies drugs, which are used in the liver cells. And if we did not have the smooth ER to make all of these things, or to do all these functions, then we would end up dying from different problems, especially this detoxifying. Long-term abuse can cause your body to produce more smooth ER, leading to drug tolerance. So if you've ever heard of people who end up becoming where they rely on a specific drug, or like they can take more meds or drink more and it does not affect them as much, part of that is because of that smooth ER that exists. Not a problem that you want to have, though. Okay, the Golgi apparatus, aka the Golgi complex or Golgi body, it goes by both of those as well. It's flattened, folded, membranous sacs. So you'll notice that these kind of all look like they're squished. And this receives packages or vesicles from the endoplasmic reticulum and modifies those proteins and lipids that were created previously. They direct them to different places in the cell depending upon where they're needed. So the function is packaging, modifying, and then basically kind of shipping them to where they need to go. Vesicle. I want you to focus on the lysosome for this because that's the main one that we talk about. A vesicle is a small spherical sac that is surrounded by a membrane. So you'll see that this right here would be the outer membrane of your vesicle. They often merge with the cell membrane and release contents out of the cell. So like this right here is a vesicle. Here's where it's merging with the cell membrane. Here's another one that's already released stuff from outside of the cell membrane. Types of vesicles include lysosome. That's the one I want you to focus on. That one will digest enzymes and it'll eliminate foreign stuff that tries to invade. And it can also break down old organelles that are no longer functioning properly so that the parts can be reused. Peroxisomes detoxify and neutralize. Glyoxysomes break down fats. We won't focus on those much, but just be aware that they exist. and endosomes that engulf material being brought into the cell. Ribosomes are small spherical organelles. Their function is building protein molecules. They do not have a membrane. So even though on our pictures they often look like they're little circles, they're little globs. They don't really have anything that contains them. Okay? Some ribosomes are free in the cytosol while others are attached to the ER, the rough ER. Cytoskeleton, and I want you to think of skeleton when you think of this because it gives structure to the cell, and that's just going to be a network of very thin tubes and filaments. You can kind of see them in here that crisscross within the cytosol. So here we've got an image. We just see a bunch of like long structures, and those are going to help give it shape, give it structure, so on and so forth. Mitochondria is the organelle with a double layer. So you'll see that there is a layer here, and then there's also another layer in here. There's inner and outer membranes as they have them labeled, which that gives it a larger surface area to allow for more reactions. So you can see this whole back and forth, that whole membrane allows for reactions to occur along the whole thing. So the function of the mitochondria is to create ATP, which is energy. And you will learn that ATP is energy as we get into respiration and photosynthesis. ATP will become a very common term, and it will use glucose to make the ATP. And if you remember from the carbohydrate unit, glucose is sugar, so when we eat sugars and starches, the mitochondria break that down and create energy out of it. ATP is adenosine triphosphate. You do not have to memorize that word as long as you know that ATP means energy. That's a big thing you need to know. Mitochondria have their own DNA and can reproduce by dividing. So like in forensics, if you could get mitochondria in a sample, like a cell with mitochondria in it, you could potentially determine who a person is based upon that. Scientists think the mitochondria originated from prokaryotic cells. The fact that they have their own DNA helps support that they could have once been prokaryotes that lived on their own as individual unicellular organisms. Chloroplasts are found in plants and algae, and hopefully you've heard of them before. They use light energy to make carbohydrates from carbon dioxide and water, primarily plants. Can't say 100% plants, but because there are some bacteria, algae that are able to photosynthesize, but very, very few. Chloroplasts contain their own DNA and are thought to descend from ancient prokaryotes as well. So think about that, the fact that both energy-making organelles are actually thought to have been previously prokaryotes, which kind of makes sense if they're able to make their own energy. All right, and we'll get into this right here. This is photosynthesis, so when we get into photosynthesis later, we will definitely be talking about chloroplasts, and that will be a big part of what we talk about then. Central vacuole is most obvious in plants. It doesn't say that they're only in plants, but they're most obvious in plants because they're so large. And they're large fluid-filled organelles, and the purpose is to store water and also enzymes, waste, and other materials. And the reason why they're so large in plants is because plants can't, most plants, I'm not going to say all, can't move to their source of food, so it allows them to survive longer without periods of water.